So in this section, I'm going to talk about PCR and genetic analysis of the soil. So for this, we can extract DNA directly from the soil. So a very small amount of soil is enough to extract DNA. This DNA is then purified and it undergoes a bit of a forensic method where we amplify the DNA. So a very small amount of DNA can be amplified to be a very large amount of DNA using a method called PCR. In this PCR, we're amplifying regions of the DNA of bacteria, the 16S ribosomal RNA region, and we're also amplifying regions of fungi, the ITS sequences. From this, we get a profile when we sequence this DNA, what kind of species of bacteria, what kind of species of fungi and other microbes are present in the soil, and to how much they are present. The quantity and the quality, which means we find out about the diversity of the microbes and also how much is there altogether and whether any pathogens may be there. This is very good to compare if you would like to do some treatment to your soil. You can look at the soil DNA before and after your treatment and see whether it makes any difference for your soil health.